Now, to introduce our speaker, I'd like to bring up the director of the Center for Mechanisms of Evolution, Mike Lynch.
I'll keep it brief because Kerry's only got 7 minutes. So Kerry was the inaugural faculty member of the CME. Took quite a risk coming here when the center consisted of just me. Done really well. Has been an incredible colleague. Helped us grow the group to now 7 faculty members, maybe 100 people or so in the center now, and helped us get a NSF center grant that kept us going for a while until it fell victim to the war on science recently. Kerry's lab uses yeast as a model system. She studies a whole wide variety of problems in eukaryotic cell biology, ranging from the use of chaperones and protein folding, what that means for cell health. Her lab has developed a whole bunch of novel techniques for doing multiplexing and of different cell populations and doing single cell sequencing of not just RNA, but DNA as well. So I think we'll probably hear some of this from Kerry directly. And all yours, Kerry. Thank you.
Thanks, Mike. And thanks everyone for being here to hear a little bit about the science my lab is doing. So yeah, I'm Kerry Geiler-Samerotte. I'm an assistant professor in the Center for Mechanisms of Evolution. And I'm going to talk to you today about a new technology for single-cell DNA sequencing that I hope is of use to some of you. So I think the heyday of DNA sequencing was in large part brought about 25 years ago by sequencing the human genome. This was a huge deal. Sequencing the human genome was reported on the front page of the New York Times. Bill Clinton called it the most wondrous map ever produced. And I think a lot of scientists believed that major medical breakthroughs were imminent. I was an intern at the National Human Genome Research Institute when this was all happening, and there was a sign in the elevator that said, "Cancer cured by 2016." And so it turned out to be a little more difficult to sequence tumors than we expected. It's easy to sequence a person's genome because most of the cells in their body possess that same genome. But it's harder to sequence tumors because tumors are comprised of many different mutant cells. We really need single-cell DNA sequencing to tease apart those mutations and determine which ones are involved in metastasis or invasive growth. And tumors are just one microbial world that we need single-cell sequencing to explore. We need single-cell sequencing to understand the diverse creatures that live in the gut microbiome. And we needed to understand the different types of mutations that can allow infections to resist drugs. Unfortunately, single-cell sequencing is always done in the same painstaking way, by isolating cells one at a time into different tubes. This is about as fun as watching paint dry, but more importantly, even if you do it with an advanced robot, you still can't get to the number of cells that we really need to understand these types of microbial communities. So I actually had no intention of working on this when I started my lab at ASU 7 years ago. My lab's mainly interested in understanding the mutations that allow microbes to adapt to new environments. But as the experiments in my lab got bigger and bigger, they started to contain more and more diverse types of adaptive mutants, and we just came up against this barrier. How are we supposed to identify the rare mutants, the rare cells we're interested in, and sequence them? If I had my way, the way my lab would do this is with toothpicks. That's the way I did it as a postdoc. I picked thousands of colonies originating from single cells into separate wells and basically hunted for the one that I was looking for. So that's how it's done even now. Like all these papers I list here are papers that are isolating cells manually. And I don't know if it's something in the air at ASU, but I could not find any lab members willing to use these toothpicks. My lab basically mutinied in the best possible way and instead created a new method to perform single-cell DNA sequencing without physical cell isolation. So their key insight is that cells are already isolated. Their DNA is isolated by their cell membrane. And the lab is using that membrane as a chamber for reactions that are typically done outside of the cell. The first step of the reaction is to amplify the DNA so there's enough of it to sequence. And that's the step that many scientists thought would be impossible, most notably the scientists who reviewed our grant application. And the reason they thought this is because the first step is to formaldehyde fix cells to turn them into sturdy containers for these reactions. It's essentially like dunking cells in super glue. So one of the problems is DNA gets essentially glued to proteins. It gets cross-linked to proteins. This makes it really hard for a polymerase to come in and glide along that DNA and amplify it. It's stopped by the proteins. We solved this by using a strand displacing polymerase. So you don't have to heat up these kinds of DNA amplification reactions to separate the strands of DNA. A strand displacing polymerase can just come in and separate that top strand, displacing it, and also displace the protein crosslinks. So when we use this type of polymerase, we're able to see really long DNA reads coming from out of our cells between 15 and 20 kb. So I think that's consistent with the idea that the polymerase isn't obstructed. Another problem with that formaldehyde glue, though, is that the cells become very difficult to crack open. And so you have to use very harsh chemicals to crack them open and get the DNA out. And it's basically a catch-22. You can get the DNA out, but now it's useless to you because these harsh chemicals are there. We overcome this by intracellularly labeling every amplified molecule with a biotin. And what this lets us do is when we crack the cells open, we can bind that biotin to a magnet-bound streptavidin bead and then wash away all the chemicals, but retain just the intracellularly amplified DNA. And these tricks turn something that may have been impossible into something that's possible. We can get it to work in very diverse cell types, including gram-negative, gram-positive bacteria and fungi. We've amplified and sequenced entire genomes from intracellularly amplified material. And we've sequenced target genes from intracellularly amplified specific regions of the genome. So our preprint is out on bioRxiv. Hope you guys check it out. There's just one small problem, and that's that was only step 1 of the process. So we still need to do step 2. After step 1, we're able to lyse the cells and get out this DNA, but the problem is now that DNA is mixed together with all of the other cells in the tube. We need a way to label it to know which piece came from which cell. Fortunately, this kind of thing is already done for RNA. My lab uses this method a lot, and we even teach it to kids at ASU Open Door. So the idea that we tell the kids is we say, how many different types of towers of Legos can you make with only 4 colors? And they usually say 4, but that's wrong, of course. It depends how many Legos you put in your tower. If you stack 2 Legos on top of one another, you can make 16 different types of towers. If you stack 3 or 4 or 5 Legos, you can make a lot of unique types of towers, a lot of unique combinations. We use the same trick for DNA. We take our millions of cells that have intracellularly amplified DNA, and we split them into 96 wells. In each well, there's a barcode that gets annealed to the DNA. Then what we do is we suck all these cells back up, pool them, mix them together, and split them back down into a 96-well barcode plate. And now a second DNA barcode will anneal. And if you do this enough times, eventually every cell gets a unique combination of barcodes labeling its DNA. And we can use that combination to trace every molecule of DNA back to the cell it came from. The caveat is that this works for RNA. We need to make it work for DNA. We're pretty optimistic. Annealing DNA to DNA is something that is pretty commonly done. And so I thought this might be a good time to share this project with all of you. We're looking for sort of test systems to see if our technology works. I don't think the best first test system is the world's most complicated microbial community, but we're interested in chatting over coffee with anyone who thinks this technology might work on their model system and seeing if we can come up with some good test cases for it. So I hope some of you are interested. Thank you for your 7 minutes, and big thanks to my lab.